Okay, we can get started. Hi, welcome to the 37th Coffee Lecture Series at the Chemistry, Biology, Pharmacy Information Center. I'm Gina Cannarozzi, and today I'm going to talk about Circos. So what is Circos? It's a tool for visualizing information in a circular layout. It's a package written in the Perl language, and the user defines the image through plain text configuration files. So it's not a point and click. The user makes configuration files, and these can have defined rules that changes the formatting. So I can say if the value is over some number, please make the picture blue. These configuration files are then compiled to produce the image. So it does involve command line, being able to work with the command line. But this is nice because this automated production can be put into a pipeline, and you can add it to your pipeline and produce the images automatically. The package was developed for genomic data, so it has a lot of functionality for genomes. For example, what I'm displaying here are grass genomes, but it's useful for so much more than that. The project was started in 2005, and the last update was 2025, so it's still relevant. Okay, why use a circular layout? Well, if your data is not confined to a grid, then a circular layout may use space better than a grid. And it also reduces edge effects that you often get with linear plots. So you can imagine a linear plot with connections in between it, then you will have a lot of crossings in the middle, and this can be reduced by using a circular layout. So what kind of data can you use with this multiple data sets with similarities that can be aligned, and that's what you see here, where I have 8 plant genomes, and they are represented as concentric circles. It actually, this kind of plot works well for plant genomes in particular, because rice here is very small, while wheat is a very big genome. And here I've aligned them such that the homologous or homologous portions of the chromosomes are aligned. So I can see very clearly that right here, there's been no rearrangements. A rearrangement is indicated here by an arrow, so I can see exactly where the rearrangements are and how each genome is related to the other one. So it's good for these comparing a lot of genomes, but you can also take a single genome, a single data set, and plot a lot of data on top of it. Another good use for this is data that has flow from one category to another, or data that has connections for one category to the other, and I'll show some examples of that. Okay, this work is the creation of Martin Gershwinski, who works in science and art. He's a staff scientist in Canada's Michael Smith Genome Sciences Center, and he does so much beautiful work on science illustration and communication. So this was an illustration he took part of in Scientific American, and he's also made a different way to visualize networks rather than this flat network view that you always see, which makes a lot of spaghetti and plots if you have a lot of things. He's developed a new way called a hive plot. I'm not that familiar with this, but I want to look into it. In addition, he contributes to Nature. There's a points of significance column that has methods on statistical and experimental design that I'm also going to look into because it looks like really good information. So seems to be a cool guy. Okay, so when you make a Circos plot, you start with an ideogram. This is the base object, and it's composed of sectors. So if we have the human genome, we have one sector for each pair of chromosomes, and then you have the X and Y chromosomes. I can do this and plot data from the human genome on top of it, or I can take 3 genomes, human, mouse, and rat, and put all of their chromosomes and then look at connections between them. As I said, it starts with configuration files. So the configuration file that defines the ideogram looks something like this, where you have an ID, a label, a start, and an end, and a color. These are predefined because for genomics, so you can change the color, but you can see that chromosome 1 has 247 million base pairs. So that's the length right here. And then once you've defined this, Circos will put them in the radial configuration. You don't have to do any kind of math to figure out where they go. Okay, once you do that, you can add tracks, and tracks are your data. So you have all sorts of tracks you can add. You can do scatter plots, you can do line plots, you can put histograms, you can plot histograms, and again, you can define rules that change the color. So here, I don't have to say make this block green or make that block red. I just define a rule that says the condition is if the value of some variable is less than minus 0.5, make it red. If it's greater than 0.5, make it green. So this kind of, every new plot that you do will have different red and green, but it's rule-based definitions. There's tiles, there's heat maps, so you can plot your expression data. And it's got so many functions, like for example, there's this variable parameter scale log base that you can change the dynamic range to emphasize small values or large values, or make everything uniformly distributed across the range of your data. So a lot of thought has gone into adding functions that will change how the data is displayed. There's also a lot of text and labels. Of course, I can add labels, but here I've put two labels onto the same place, so it automatically makes this little fork and does it. And if I stack the labels, then it will find the best configuration that uses the space in the best way. So here I've labeled all of the genes on this chromosome, and it places them and does these little red bars that show exactly where they go. Again, you can have rule-based labeling. So here the font size is changing depending on how close it is to 5 here. So you can define any kind of rule you want. There's symbols, bubbles, connectors, and I don't have to put the connector. I just put the positions and it will draw the connector itself. You can stack the plots, you can change the radius, you can put different kinds of data on different segments here. Okay, so that's how you put data on the plots, but the other really nice feature of this software is that you can use links to show connection and to highlight flow. So here we have the human genome, and you can see I've plotted two data tracks on the outside of the ideogram. And if you look at this, you can see that what's being plotted here is like density of size and copy variance, and you get a good view that here it's less dense and here it's more dense. So you can look at the big picture of what's going on in your genome. And here the gray lines are genes in the same pathway. So what you highlight is limited only by your imagination. And here the colored lines show similar regions. So you can see maybe at some point there was a duplication here and the genes here got distributed across from this chromosome that was duplicated. These genes have been duplicated, but all ended up here. It's also interesting to me that you have some kind of duplication here and it only shows up here and here, but not anywhere else. So that's the way to look at the big picture of connections between genomes. And I can do that also with two different genomes. Here you have the donkey, here you have the horse. They don't have the same number of chromosomes. And interestingly, chromosome 4 in the donkey is red. I've made the connections red so I can see what happened to the donkey chromosomes. And this one seems to be split between chromosome 28 and chromosome 19 in the horse. So that's a comparison, the same kind of thing within two genomes or between two genomes. But you don't just have to It's not just used for genomics. You can visualize any kind of flow. Here we have human migrations between 2005 and 2010 from this science paper. And you can easily see there's a big migration between Southeast Asia and Western Asia. I've colored it on the people that are moving. I mean, not I've colored it, the authors have colored it that way. And you can see, for example, here, a big migration from South America to Southern Europe during this time. It's been used to track customer behavior and car transactions. So if I leave the kind of car I have, where do I go? And even used to analyze how many times names were mentioned in political debates. This is from 2008, and you can see eventually Obama got the nomination, but during the debates, the Republicans at least were talking more about Clinton than they were about Obama. So it's good for these kind of visualizations. I want to also mention Circlize, which is an implementation of a similar package based on Circos, inspired by Circos, but it's in R. So if you're an R user, you can use this. And it has also a phylogenetic tree, which might be useful. And so that's it. What I like about this package is it made me think about the best way to display data. So when you have data and you need to plot it, it's always good to think, what is the best way to represent that? Would a circular plot be useful for me? Maybe if you have data with flow or state changes, consider Circos for visualization. It requires it's not easy to use. I would say it takes some command line experience. I did have trouble installing it on the Mac because the Perl libraries wouldn't install easily. So that's it for Circos. Are there any questions or questions online? Ooh, I stopped it. Nope. Okay, then thank you very much for coming, and we'll see you next week. Oh, I forgot to say it. Let me show what's coming up next week. Will be coffee lecture card for today. And next week we have VOS Viewer to visualize literature networks, Marcouche structures, and altmetric. So see you next week. Okay, bye.